This video covers the most missed topics from the Unit 1 exam. Part of why I'm going over this is because it's always good to make sure that you understand key concepts, and also these key concepts, these topics, are going to be important as we move through the course. So here are the topics we're going to talk about: the characteristics of different types of microbes, the scientific method, opportunistic versus true pathogens, digestion, vaginal versus cesarean birth, interpreting graphics and papers, and sensitivity to antibiotics. So first off, why are the characteristics of different types of microbes important to later topics? Well, because these characteristics are targets for antimicrobials. That's one of the most important things. Also, it helps explain how they interact with each other in our microbiome. So I've made a video, one that I intended to make at the beginning of the semester, and I had technical difficulties. Well, now it's done. I'm putting it in week 6 as required video watching, and I'm also putting it in week 1. So that's where you'll know where to find them. Next up, let's talk about the scientific method and why it's important to later topics. The scientific method is how we figure out all of this stuff about the microbiome, and with some of the things that you're going to read, it's going to be talking about the results of this process. So it's important to know what the scientific method is. The things that were missed the most from the scientific method were the terms theory and hypothesis. So let's go over these specifically. So in scientific terminology, a theory is a testable idea with predictive value. In other words, we can use this theory to predict the results of experiments, and it's supported by a boatload of evidence. In non-scientific usage of the term, it means an untested idea, which is exactly the opposite of how we use it in science. So it's understandable that this is confusing, but make sure you remember that when I use the term theory, and when folks that are presenting information in the videos and in the reading material that I assign to you, this is how we are using this term. We're not using it in this way. Now let's go to hypothesis. In science terminology, it means a testable idea that may or may not have been actually tested. So in other words, a hypothesis becomes a theory. So a hypothesis is kind of what we should be using in non-scientific terminology, but we're not going to be able to change that. So let's talk about the difference between opportunistic versus true pathogens. Why is this important to later topics? The difference between an opportunistic and a true pathogen is very important in understanding the microbiome. And it's confusing. Don't feel bad if you don't have it down yet, because the scientists are still figuring it out too. There used to be no question about whether something was a pathogen or not. It was just whether it was a pathogen that caused severe disease. So opportunistic pathogens usually caused mild disease, only caused disease in immunosuppressed individuals. Now we've changed our minds. So let's talk about the current understanding of the difference between an opportunistic and a true pathogen. So first off, does the microbe ever do any good within the human host? If the answer is yes, it's an opportunistic pathogen. If the answer is no, it's a true pathogen, and it's really not part of the human microbiome. At least it's not a part of the human microbiome in health. Now let's talk about numbers. If an organism is a long-term resident and it's in low numbers in health, or if it's a long-term resident in high numbers causing disease, it's an opportunistic pathogen. If it is active and transient disease, or inactive, in other words, it's hanging around in the host, in latency, then it's a true pathogen. Yeah, that's confusing. Let me give you some examples. E. coli, generally speaking, hangs out in all human intestines. You can't get away from it. It is considered a resident. And if it's in low numbers, you're healthy. And it does some good. It produces vitamin K for you. So it is definitely an opportunistic pathogen, because if you get high numbers, it has been correlated, not proven to cause disease, but it is correlated, or it's highly suspicious when it's found in disease cases. Now, here's an example of a true pathogen. Cholera is never found resident in the human unless it's causing disease. It's a true pathogen. Here's another one. TB can be found in active state, causing active tuberculosis, or it can be walled off and sitting around waiting for better times in inactive tuberculosis. In neither case is it causing any good, so it's a true pathogen. And let's move on to strain variation. You can have an opportunistic pathogen that moves to a true pathogen because it has picked up antibiotic resistance, a toxin, all sorts of good stuff. So an opportunistic pathogen may be a true pathogen depending upon the strain. Confused? So are the microbiologists. Although we're getting it worked out. Actually, there's some microbes that we've already got nailed down, but there's some of them that we're still figuring out their true relationship to the humans, to the human host. Now let's talk opportunity. Opportunistic pathogens cause disease when they can. When they can't, they're team players. Pathogens, on the other hand, will always cause disease. Either that or the immune system clears it out, or your skin keeps them out, or whatever. Hopefully that helps you with the difference between opportunistic versus true pathogens. Let's move on to digestion. Why is it important for later topics? Because it helps us understand the relationship between humans and their microbiome, the microbes that we play host to. So, first off, let's talk about enzyme production in humans and enzyme production in microbes. In humans, we produce a lot of digestive enzymes in the small intestines. We produce enzymes that break down proteins, lipids, carbohydrates, all sorts of good stuff. In the large intestine, so far as we know, we do not produce any enzymes. We digest the food that we can in our small intestines and then absorb that in the small intestine. But microbes, okay, in the small intestines, you do have microbes in the small intestines, and they are producing enzymes to get their shot at the food. But there's even more in the large intestines, for the reason being that whatever comes through is fair game. They're not competing with us to break down food and to get it. So, whatever gets broken down in the large intestines is being broken down by microbes. So why do we care? Bifidobacterium, Bacteroides, some of our mutualistic symbiotes that occasionally become opportunistic pathogens, produce enzymes to break down complex carbohydrates that we cannot. So if you have a good number of these, you're going to break down more of these molecules and you're going to have more calories available to you. If you're living in a sustenance level farm where you're hungry a lot of times, this is a good thing. In the United States, maybe not so much. We'll talk some more on the microbial influence on obesity. Let's go to breast milk consumption as adults. I had a question on the test asking if you wanted to stimulate your bifidobacterium population within your gut as an adult, what would you consume? And a lot of you picked breast milk. Well, let's talk about why I consider that to be an incorrect answer. First off, as adults, you eat a large variety of foods. So you can eat vegetables that have complex carbohydrates as an adult that you couldn't eat as a baby. Also, you've got a complex community of microbes in your stomach and in your intestines that you did not have as a baby. Therefore, it is my contention that human breast milk would not change the population significantly in an adult. Whereas in a baby, since that's all they can eat because they don't have teeth, they're not producing as many enzymes as the adults are. Therefore, it's going to have a greater effect in babies that are starting their colonization process of their friendly microbes whereas in adults it's not. Also, I wouldn't want to take milk from a baby in a NICU, so I'm not going to run out and go buy human breast milk. Sorry. So I would eat more vegetables instead. That's me. So let's talk about the microbial influence on obesity. So far, we have found that there are two things that are influencing this as far as the microbes. There are microbes that are digesting the same food items to create more available calories, that if those microbes are not in an individual, or if they're in lower numbers, that individual has access to fewer calories. That's going to influence fat deposition or obesity. The other set of microbes cannot break down high carbohydrate foods, but they do like simple carbohydrate foods. And so they can create cravings for these high calorie foods that both those microbes and we can get a large number of calories from. Yes, microbes can talk to your brain. They can send up a signal saying, oh, I could really go for a donut right now. Now, so far as I know, we haven't figured out which microbes are causing these cravings, although there are some that I have suspicions about. But we're going to wait for the scientific community to find this out in more detail. Let's talk about vaginal versus cesarean birth. Why is this important to later topics? Because how you're born influences your start on your microbial community. And we're finding this influences allergies, asthma, obesity rates, diabetes rates, cancer rates. It goes on and on and on. So knowing how the birth process affects the start of developing your microbial community is important. So let's imagine the process. Here's a cesarean birth. The baby is lifted out of the mother without going through the vagina. It's all very sterile because you don't want microbes getting into the abdominal cavity of the mother. So the gloves are sterile, everything's sterile, their baby's put into a sterile blanket. But after that, what do? You kiss them, you touch them, you hold them, you pass them around. So the microbial community of babies that are born by cesarean are more like the microbial populations of human skin. Makes sense, we're touching them. With vaginal birth, on the other hand, the baby comes out the vagina, which in Missing Microbes you've read about how that community changes in preparation for the birth of the baby. And so the baby swallows a lot of lactobacilli, and also the baby is exposed to the mother's intestinal contents. Now, the joke I use is that back in the day before hospital births in enemas, the baby would land in the mom's pile of poop. That's not necessarily true, but it helps with the visual image. So when we test the microbial communities of babies that are born vaginally, their microbial communities look more like the mother's vaginal and intestinal communities. We can seed their skin with skin bacteria. You're supposed to hold and kiss and touch babies. But you want intestinal bacteria getting into the intestines of babies. Now let's talk about interpreting graphics in papers. Why is this important? I'm going to be having you read some papers. And it'll be important to be able to interpret the information in there. Also, you're probably wondering why I keep bringing up the article on irritable bowel disease and Crohn's disease and some of these other things. This was the article that I read that got me interested in the human microbiome, which led to this class. So of course I keep going back to it. So let me pull up the graph from the IBD article and let's talk about how to read it. So let's take a look at this. How the authors have set up this table is that for this part, this is talking about colorectal cancer. This part here is talking about Crohn's disease, and ulcerative colitis is over here. See how they've color-coded it? So the purple is with the purple and the orange is with the orange and so on. And so here they're showing that when there is an increase in numbers of E. coli, methogen-producing bacteria and sulfate-reducing bacteria, you see this increase in ulcerative colitis. Does it cause ulcerative colitis? We don't know that yet. In ulcerative colitis, we see fewer numbers of Bacteroides, Bifidobacterium, Faecalibacterium, and Rhinococcus. We also see an increase in Lactococcus species and a decrease in Clostridium species, which is kind of odd. We don't see that in the other types of diseases. We see an increase in these types of organisms in colorectal cancer. We see a decrease in these types of organisms in this same disease state. Does this cause the disease, or is it just a result of the disease process, say maybe other processes that are going on? We don't know yet, but it's really suspicious, which is why we're doing more research into this. So we've got the same thing that we're looking at here. So in Crohn's disease, we see a decrease in these. We see a decrease in some Clostridium species. We see an increase of others, and we see an increase in these. So hopefully that helps you interpret this table, because guess what? I'm going to be talking about it some more. Here is our last topic. I had a question where you were supposed to match different microbes that we'd covered in the microbe of the week to their sensitivity to antibiotics. And as it turns out, everybody missed that one. It was a poorly written question. Sorry about that. But let me go through what I thought your thought process would be, and be reassured that I gave everybody points for that question, because like I said, it was poorly written. So what I was hoping is that under the very sensitive category, you would put genera of bacteria that were sensitive to cell wall inhibitors, sensitive to protein synthesis inhibitors, and sensitive to folic acid synthesis inhibitors, as opposed to sort of sensitive that are sensitive to only 2 of these classes of antibiotics, as opposed to not sensitive at all, which are your endospore formers, eukaryotes, and when we get to them, viruses. That's what I was going for. Was it obscure? Yes. Especially when we go into talking about antibiotics and how these organisms can pick up genes that make them not sensitive at all to, say, cell wall inhibitors. There's a lot of gram-positive bacteria that are able to do that, and we will be talking about that in week 6. So I hope that that helps you get a handle on some most missed topics from Exam 1, and hopefully it will help you as we go through this next unit. Let me know if you have any questions.